So I try to cover, so my assign is the design aspect, but some of the repetition has to be there. So I still want to give some quick overview at the first module, and which basically give what is phase 1 trials and some more maybe relative specific design considerations. But then I want also touch a little bit about different types of phase 1 trials. One is more on the treatment mechanistic trials, and which is turning to be more and more interesting with biological treatments and products, and also target therapy. And tomorrow, Dr. Kamen will also emphasize on that. And also then I will talk about more about traditional dose-finding trials. So we know there's a clinical trial or drug development process as a staging process. There are different components. And when we put phase there, the purpose is trying to characterize the stage of development more easily, so we have a common language to talk. And so when we talk about first in human trials, and Spiker in 1991, there's an old book that basically frames that as phase 0 and phase 1. But it's more mixed. Sometimes pharmacodynamics and pharmacokinomics also, kinetics studies also in phase 1. So it's the characteristics of that, of course, usually it's a new drug or new treatment, and usually very small sample size. And there's no hypothesis developed there. So the idea is really exploratory. And so for first time in human, you want to make sure, understand their toxicity and what are the organ systems that really involved. And then you establish also optimal biological dose, understand the PK pharmacokinetic parameters, and also what is tolerability and the feasibility for the medication. And rarely, but increasingly, particularly with target therapy, we also want to look for the evidence for efficacy. And I will mention also later on about different type of dose-finding study, that combination of looking for both efficacy and the toxicity. And also, we want understanding that PK and PD relationship. So it's very important of phase 1 trial. People, well, not only it's because the beginning of the interaction, put the drug into the human, but also it's set later on the stage in development. And in the 2012 talk by Kelly and Vinok, that they look for the combination therapy in patients. And what they find out is when they combine the agents, and the patient experience higher toxicity, and most of them cannot complete the treatment cycle. Therefore, you don't have a chance to benefit from the efficacy. Well, because the combination may not have been through properly for early phase trials, so the dose may be not right. And another case, of course, is acupuncture. There's no safety concerns. But surprisingly, there's no phase 1 trial for acupuncture, that how much exposure time, treatment time, actually will lead some signal or minimum effects. So there is a trial that reported in JAMA last year, and it created a lot of concerns. It's compared acupuncture with usual care and on the chronic knee pain, and the conclusion was a negative trial. But if you look at the protocol, the patient only received 20 minutes every 1 or 2 weeks of acupuncture, so the intensity is not there to cause any difference. Again, there's no early phase to support the dose of choice. So, and also I want to mention also phase 1, phase 2 is phase 0, phase 1, phase 2 is kind of a turn. It's not always clearly cut. And there are also translational pieces. You can view it phase 0, you can view it phase 1. But also there are increasingly what we call seamless phase 1-2 trial, that for the later stage, you just keep the patient in phase 1 and start to evaluate their efficacy component. And also, it's best opportunity for university investigators that to be involved in this kind of early stage, because you may have more knowledge about the innovative agents that from your lab and so on. So the 2 types of phase 1 trials. The one is more on the treatment mechanism, and usually carried by not only company but also at the university level. And it's increasing importance because with target therapy. And the idea here is that not only you want to, before you really expose high level of full trial, you want to see if the target actually respond to your treatment. So you want to put the drug, usually it's smaller sample size, and the target, so we will, and the target is in the biological level. And this is related also is what increasingly they're called biomarker-driven pharmacologic audit trials. There is a paper that, a review paper I will mention later, and also I think Dr. Kuma will mention that tomorrow too, that oncology trials that the new drug will necessary to do that phase. And the other part is more traditional phase, is dose finding and look for human pharmacologic characteristics, and also develop safety dose. And the goal, of course, is to find the right dose to recommend to phase 2 trials. And there's also a paradigm on the diagnostic tests that Dr. Goodman is going to mention later today. They have also early phase and later phase piece. So in design considerations, and usually depends on type of drug, and most of non-toxic or low toxicity drug that people put normal volunteers to do the test. However, for cancer or invasive, the validation need to be invasive, and usually they go to refractory late-stage patients. And so it's very critical that you want to make sure who, as Rita mentioned about population, who you want to expose to the treatment. You need to establish clearly eligibility criteria. And there are two ethical concerns here. One thing for the non-toxic drug, because the reason that you want expose is there's a risk to induce unsafe adverse events, right? So you want limit exposure of healthy normal volunteers to the harm. And also for patients that, even they are patient, you don't want overdose them to unsafe dose. But the other side of the token for particular oncology trial, because these patients are in the later stage and infracture disease, you don't want to put them very low dose because there's no benefit to the patient. So in the design phase, when we say a good design, you want to make sure those patients exposed to the MTD, if possible, as many as possible. And there's no randomization involved in the phase trials. It's always open label. And the selection time points and also the statistical models, because a lot of those exploratory based, they're non-parametric approaches, but because small sample size, it's sometimes more efficient if you have model behind. And so therefore, that's 3 components are associated.